Welcome to the Science Edit, the podcast that breaks down the trickiest parts of science and helps you get it right when it matters most. I'm Miss Lima, and I'm here to support you with the science you need when you need it. Whether you're revising for exams, exploring future careers, or just trying to figure out why your last test didn't go so well, you're in the right place. Let's dive into our next episode. You've revised, you understand the content, and you feel like you've written a solid answer, but somehow the marks still aren't coming. In this episode, we're looking into some of the most common mistakes students make in GCSE Biology. From osmosis to enzymes, inheritance to food tests, we'll break down where things go wrong and how to get them right. If you're aiming for full marks, this is the episode to listen to. Why do I keep getting it wrong? The biology basics. So I'm joined today by 3 Year 13 students who've been through this process. Thank you so much for joining me today, gentlemen.
More than welcome to be here, Miss.
Thank you, Miss.
Thank you.
So the first topic we're going to discuss today is movement in and out of cells. So diffusion, osmosis and active transport. This is such a large topic for GCSE Biology. How did you approach your revision for this?
In terms of these processes, it's all really, for me, it was a lot of memorisation, and it was also sort of pairing up and finding the differences between all of them. Making sure that you know the differences between these three different transports will make it really easy to remember which ones do which.
Yeah, definitely. It's finding those differences that allow your brain to remember why certain substances move by specific ones. The key thing is making that distinction in your head. And once you've got all three, then when you go into a question, a lot of times when it's application, then your brain will automatically snap to one of them, and then you'll be able to talk about it and go through all the processes you've memorised.
Yeah, for example, remembering active transport happens with ATP, so it uses energy, whereas both of the other ones do not.
Yeah, they're both passive processes.
Yeah.
When I tell students to revise for this topic, and it comes up in every paper, it's such a cross-topic area, what I say to them is memorise your definitions. That's the first stop.
100%.
Memorise your definitions, and also in a question always state the direction of travel. So if you're talking about osmosis, where is the water going from and where is it going to, and make that really clear, and then say from a high water potential to a low water potential.
Yeah, I think that really helps because each one has a different distinction. So for active transport, you might have low to high because you've got that energy, and that's where those application questions come in, because the examiners will want you to figure out which one it is, and if you know your definitions, it's an easy way to pick out which mode of transport it is.
So, as Miss Lima said, sort of with the specifics of the question, such as knowing what moves, so with osmosis, it's the water moving across the partially permeable membrane. It's not what's dissolved in the water, it's not any of the other substances, it is the water moving. And then being specific with direction of transport, as you said, but also what might be moving in the specific case, such as active transport in plants. Mention the ions and anything else that may be taken up by the roots.
The other thing to remember is if you're dealing with cells, then osmosis in plant cells as opposed to animal cells, very different, because plant cells have the cell wall, so cells become turgid, whereas animal cells will burst. So that's a really important fact to remember, because it often is the point of the question. So the next topic that we're going to look at is enzymes. So what kind of things do you need to remember to get full marks in enzyme questions?
So you'll need the basic definition of an enzyme. So we need to mention how it speeds up a reaction by providing a route of low activation energy, not simply decreasing the amount of energy for a reaction to happen. And that it also does not get used up in a reaction and can be used again and again and again for further reactions.
You would also have to remember where each enzyme is produced and where each enzyme acts. For example, amylase is produced in the salivary glands and it acts in the mouth and small intestine.
Yeah, I think for me when I was revising this, it was drawing out, simple outline of a person and then drawing on where they're produced, where they move to, colour coordinate each enzyme and then draw arrows to where it goes. Then you can draw annotations off and say what it breaks down, why it's used, any specific cases where it might be used, if there's certain conditions that have, some of the enzymes might be affected by.
Pepsin, for example, with pH of 2 because it's produced in the stomach.
Exactly, yeah.
Optimum pH of 2.
So have that kind of complete overview of all the enzymes, and that will help you piece it together in your exam.
I do think that a really important aspect to make sure you're strong on, because enzymes, again, it's such a cross-topic area, is to be strong on your kinetic theory. Make sure you're talking about collisions. So if you're increasing the enzyme concentration or substrate concentration or even temperature, that you're talking about, number of collisions, number of successful collisions, and that's going to increase the rate of reaction. Then also link that to an increased number of enzyme substrate complexes, making sure that you use the term complementary. So,
Definitely.
actually, it's often worth a mark just to say that, the substrate is complementary to the enzyme. When you're talking about denaturing, you talk about the active site changes shape, and exam boards are really picky with making sure that you actually include that. And don't forget as well, with pH, that will also change the shape of the active site, whether it's a high pH or a low pH. And I think some students just think, acids will denature the enzyme, but don't realise that alkali will as well. So that's also another really important thing to remember as well.
I think another important aspect about enzymes is knowing how to read an enzyme graph. For example, when an enzyme actually denatures due to the change in temperature and pH. Also when and how to distinguish between what is the limiting reactant in the reaction.
How to identify limiting factor, where you get that plateau on the graph. Another common topic where students lose marks are inheritance diagrams. Now this one's a really interesting one, because what you tend to find is most students can look at an inheritance question and if they can understand the basics between, for example, the recessive and dominant alleles, then actually they're able to draw their Punnett square quite easily. But even when they're getting the right answer, they won't get full marks because actually they haven't broken it down. So what kind of things do you need to remember when doing an inheritance diagram?
I think not specific things, but the key thing is I had to do these questions over and over and over and over and over again. I remember sitting in lesson, Miss would say, grab the whiteboards, we're going to draw a Punnett square, and then you have to write out your explanation, and then we go to the mark scheme and you compare, have you got everything for the mark scheme? Have you got all the keywords? That's linking back to knowing your definitions. And then it's kind of looked over, but laying it out in a way that the examiner can clearly see that you know what you're talking about. And I think that will really help you gain marks a lot faster, because you can write out, you can deduce a formula, find a way that works for you, and you can lay that question out same way every time, change the numbers or the letters that are given to you in the question, and that will really help you pick up those larger mark questions.
I can remember vividly at GCSE Miss drilling into me what a Punnett square should look like, especially with sort of some of the silly sort of things. Well, I say silly, but it's all important so you get your marks. It was drawing the circles around your alleles in a Punnett squares. That was one thing I'd always get and be like, you're missing easy marks. And this, we've got to remember, some of these questions, they may be big questions, but even if you can get a couple easy marks, that will go a long way over the course of your exams.
Punnett square question will generally be worth 4 marks, and you'll get 1 mark for identifying the correct gametes. You'll get 1 mark for putting them into the Punnett square correctly. You'll get 1 mark for then drawing out the genotypes of the offspring and linking that to their phenotype, so actually writing that into the Punnett square, and then 1 mark for getting the correct answer, for getting the correct ratio at the end. So I think lots of students think, I've got the correct ratio, I'm going to move on. And actually they potentially could have lost 75% of the marks on that question. Yeah. So it's really important to remember that. Just moving on as well, because another common exam mistake people make are between describe and explain questions. Has anyone got any tips on how to approach those types of questions?
So explain is saying the scientific processes of what is actually happening, whereas describe is when you say what you see.
Yeah, and that could be in a table or in a graph, looking at the shape of a graph maybe as well. Usually with describe, if you've got a graph to describe, there's often 2 or 3 marks even on that. Has anyone got any tips on what to put in a 3-mark describe question?
I think you'd have to state the overall change in what's happening in the graph, then pick out specific points and what happens at these specific points and in between these points. For example, if the overall change is increasing, but then between 10 or 20 seconds it's fluctuating, you'd have to say that.
That's worth 2 marks. And then the 3rd mark would be, I always say, manipulate your data, put some data in there, not just citing it from the graph, but actually, say, it's got 3 times quicker, 3 times larger, or there's a 40% change. That would generally be worth the 3rd mark. We haven't really touched on core practicals yet, but there is an osmosis core practical and an enzymes core practical. But one of the core practicals that students really seem to fall down on is remembering their food tests. I just wondered if anyone has any tips on how to remember those colour changes and those methods.
I remember some of the things I would do, and I still do it to this day for other subjects at A-level, is sort of making up silly little ways to remember them. I'll sort of combine the words and make absolutely outrageous links between them and other certain things just to sort of work its way into my brain. And that's just how I remember things. Because, of course, you can always write it down and drill it into you. But if you can find a way that really, really works for you specifically, then go back to it, keep on using it. Because if that's what works for you, that's what's going to help you. That's what's going to really help you in the long run.
Yeah, I think similarly to that, when you come up with that first link, whether it's, I don't know, I think for me it was the first letter of the type of chemical you're using for the food test and then the colour change. Once you've made that link, don't change it, because that's the first thing your brain has thought of and that's the thing that's going to stick.
I've never realised that before. Benedict's, blue.
Yeah, that's
Biorette,
exactly that.
blue. I've never actually realised that before.
So that's how I linked it. The way I did it was you write them all out, nice and big on a whiteboard or in your book, and then literally look at it as a whole. Look at it as if you're looking at a picture and just find things that work and link. And for me, you've got space around the question, quickly write it out, make it obvious to yourself before you've even read fully through the question, see what the question's asking you, but then write out your food tests. Because if you've got those laid out, you're now not able to hold that in your brain as much. You can refer back to basically a sheet which gives you the answers. So if you can get those down on paper, that is really important. The other thing is, for me, it was writing out the method. Get a flashcard that says, find a piece of paper and write out the test for lipids. And when you come to that revision card, I use revision cards a lot. You can use them, on the way to school. You can use whenever. So they're a really good way of revising these kind of things because it's all recall and they're easy marks in the test.
I think the method is really important to memorise. For example, a commonly missed part of the method for the Benedict's test is that you have to put it in a hot water bath. And, we're talking 65 degrees plus. Lots of students miss that. It's such a simple thing. And it will cost you a mark, just forgetting that. So it's just, and food tests are going to come up probably because they are, a key core practical.
Yeah, I think, especially if you've got something like a hot water bath, you can think, okay, so it's for the test for sugars. When I eat sugars, my hands get really hot. Oh, okay. So, yes, it's not a direct link, but now in my head I'm thinking, whenever I'm thinking sugars, also sugars, the test goes 3 different colours. You can think sweets come in all different colours. You can say that as well. Your brain's now going to think, oh, my hand gets hot. Why is that? Water bath. That's what I need to remember. And you can make those kind of links.
What you also need to remember is the different equipment you need to use for the different tests. For example, for Benedict's solution, you'd use 2 centimetres cubed of Benedict's and 2 centimetres cubed of the sugar you're testing. And you'd also have to put it in a hot water bath. And for the iodine test, you'd only use a couple drops of iodine and use a spotting tray.
So looking at what top tips you could give to students when approaching their biology revision, what's the one tip you would give them?
I think for me it was kind of getting a full view of the whole course and kind of having a perspective on, okay, I've kind of got an idea of what I need to try and learn, and make it clear to yourself what is recall and what isn't. Because for me, your teachers have been teaching it probably 10, 15 years. They're going to know everything. You're not going to be able to memorise absolutely everything. So find those things that come up in the mark schemes with the direct answer, whether it's a genetic variation question and it has the same sentence to start it every time, or whether it's a food test, you write the same thing down every time. Find those things, write them out on a piece of paper, and then when you've learnt it, tick it off, and then keep retesting yourself and make sure you know those. Because when you sit in that exam and you find the first 5-6 questions are all recall, it makes the rest of the paper so easy because you've got those marks. And in the end, it's all about just getting the number of marks you need to.
For me, specifically with biology, I'm sure Miss will attest to this, it was being specific in how I answer questions. Because I'd always think, I've written down what the question wants and I've done the meaning of what they're looking for. I hadn't been specific. I hadn't done keywords or sort of matched it to the mark scheme. So I had the gist of it, but I hadn't answered it fully, and that's what really kept me back from pushing into top marks, which I had luckily fixed by the time I came around to my exams. And it can't hurt in biology. Revision and recall will go a long way, because if you know it for one thing, then a lot of the times it's all interlinked and you can apply it for another. And get into the habit of knowing those key words just goes a really long way.
What I also found really helpful was just doing them past paper questions, because then you'd actually see what you've done wrong and actually learn the mark scheme, so you're getting them top marks each question.
My tip is actually on everyone's least favourite aspect of biology exams, I would imagine, which is longer answer questions. You know, those 6 markers that you get, the evaluate questions or compare. So, for example, if you've been given some data on heart transplants and artificial hearts and you're being asked to evaluate those, then actually what a student will do is they will often just talk about the heart transplant and then talk about the artificial heart, and they won't actually be comparing them. So it's much better to actually lay that out and saying like advantages and disadvantages rather than heart transplant and artificial heart. And I think that's really good, like actually having similarities and differences as your subtitles, and not forgetting as well that it's actually perfectly acceptable to write either a table or your answers in point form. You never get penalised for not writing in, full sentences. So that's something that's really, really important.
Yeah, I think I didn't even believe it when it was first told to me that you could have these 6-mark questions, which in my head were mini essays in a sense. It was, okay, I've got to think of my points, I've got to link them all together, I've then got to compare. No, it's biology. Get the points down, make it clear. The examiner's going to sit there and they're going to be the happiest person in the world when they see you've written advantages. They're going to go tick, 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 disadvantage, tick, 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 comparison. Easy peasy, there's your marks. Don't stick to that kind of long answer, because you won't get to the point. No matter what you do, you're not going to be able to connect it as thoroughly as just putting those points down. That's what the mark scheme is. The mark scheme is just a list of points. So if you can replicate that, then you'll get the top marks.
I think that's really valid, actually, that lots of students think, oh, I wrote loads, so therefore I'm going to get some marks. It's not necessarily true. They're looking for those key terms.
100%.
Well, thank you for listening to this episode of The Science Edit. We hope it's helped you to spot some of the common pitfalls and feel more confident about how to tackle biology questions in your exams. If you've got thoughts about this episode or ideas for future ones, we'd love to hear from you. Just drop us a message at contact@thescienceedit.com. See you next time.
Thank you.
Thank you.
Thank you.